This presentation is about different antigen formats that can be used in T-cell activation assays. Antigen is a collective term for any matter that can trigger an immune response. For the purpose of this presentation, antigen means matter that can trigger a T-cell response. We will explore the use of antigens in T-cell assays, discuss advantages and disadvantages of some antigen preparations, and consider useful assay controls. We will also discuss a new technology for producing specific assay controls called TERS. T-cell assays may utilize antigens from a variety of sources and in different preparations or formats. Let's run through some of the characteristics of such antigens. Whole or partial antigen lysates from viral or bacterial sources provide a multitude of potentially stimulating peptides, but may contain impurities from culture and expression systems, for example, endotoxins. Proteins may be complete proteins or large protein fragments and may exhibit a complex or simple conformation. They will provide many potentially stimulating peptides, but may also vary in purity and endotoxin content. Peptides provide as many potentially stimulating opportunities relative to the number of peptides used. They may be of variable length and purity and may carry native or modified C and N termini. Peptide pools may vary in terms of any of the characteristics of peptides above, and additionally, in pool size. Protein-spanning peptide pools, in particular, may vary with respect to the overlap between peptides, or the absence or presence of additional peptides. A T-cell epitope is the antigenic determinant recognized by the T-cell receptor, or TCR, of a CD4 or CD8 T-cell, for example. Such T-cell epitopes are comprised of a peptide presented in the context of an MHC molecule. The binding of the TCR is stabilized by the binding of the accessory molecules CD4 and CD8 on so-called T helper and cytotoxic T cells, respectively. Note that these names do not mean that, for example, CD4 T cells could not be cytotoxic as well. This is actually quite common. Note that T cells recognize antigen through the T-cell receptor, or TCR for short. TCRs are found on the surface of all T cells and determine their antigen specificity. All T cells are specific for some antigen, but the TCRs are generated by a random process called TCR rearrangement. This process rearranges TCR coding gene segments in such a way that a random combination of available segments are joined together. In this case, VJ and VDJ for alpha and beta chains, respectively. This is the main reason for TCR diversity. Once an antigen-specific T-cell has encountered its cognate antigen, it undergoes initial activation. Depending on the circumstances of this activation, the T-cell becomes an activated, antigen-experienced T-cell, or it may die in activation-induced cell death. The most efficiently activated T-cells will begin to proliferate and, as a result, increase in number, while non-antigen experienced or naive T-cells circulate at very low frequencies, estimated in the range of 1 in 10,000 to 1 in 100,000 and less cells. Following antigen-induced proliferation, frequencies can be much higher and will typically be in the order of 1 in 1,000 to 1 in 100, or 1%, but sometimes can be as high. There are assays for antigen-specific T-cells that depend on T-cell activation, and others that can detect T-cells irrespective of their activation state. T-cell activation assays detect activated T-cells only. Activation assays may use one or more activation markers. Using more than one activation marker increases the chances of detecting activated T-cells, because not all T-cells upregulate the same activation markers. The most commonly used single T-cell activation marker is probably interferon gamma. Combinations often include interferon gamma, tumor necrosis factor alpha, or TNF for short, and interleukin-2, or IL-2 for short. But other markers are used as well. Note that different types of T-cells will upregulate different activation markers. While CD4 T-cells may often be detectable by IL-2, this marker is not displayed by a majority of activated CD8 T-cells. T-cell activation assays are often based on flow cytometry. Activated T-cells can be detected by flow cytometry when a fluorescent label is attached to an activation marker either on the surface of the cell or inside the cell. The standard flow cytometric test is based on intracellular cytokine staining, or ICS for short. ICS works by inhibiting the activation-induced release of cytokines or other mediators, and then staining these mediators inside the cells in which they were produced. For example, interferon gamma can be stained in T cells activated by a viral antigen. The T cells producing the interferon gamma can be specific for that virus. Usually, cell suspensions containing T cells are stimulated for 6 to 16 hours, washed, then stained on the cell surface, fixed, permeabilized, and finally stained intracellularly. The fully stained cell suspension is then analyzed with a flow cytometer, and, depending on the number of parameters stained, T cells or T cell subsets can be selected. Activated T cells can be identified and enumerated. An alternative to intracellular staining following activation is the so-called cytokine secretion assay, where T cells are stained by a bispecific antibody on the cell surface prior to activation. The bispecific antibody recognizes a molecule found on white blood cells. Following activation and secretion of the cytokine, it is then captured by the capture antibody and subsequently counterstained with a fluorescently labeled antibody that also recognizes the secreted cytokine. This detection antibody carries a fluorescent tag, which is why activated cells can be detected by flow cytometry in the same way as cells stain intracellularly. ELISpot is an alternative method using a very different platform. Unlike with flow cytometry, the T cells upregulating activation markers are not secretion inhibited, and they are not present when the assay is finally read. During activation, T cells typically sit at the bottom of a 96-well plate and secrete cytokines like interferon gamma and other activation-induced mediators. The plate is coated with a capture antibody recognizing interferon gamma, for example, and the secreted interferon gamma is captured following its release from activated cells. All cells from the plate at the end of the incubation time and the bottoms of the wells are coated with a solution containing a detection antibody recognizing interferon gamma, albeit a different epitope than the capture antibody to avoid competition. The detection antibody is labeled with either an enzyme or a fluorescent marker. When the ELISpot plate is read, cells secreting interferon gamma will have left a footprint or spot on the plate's bottom whose intensity and size correlates with the amount of interferon gamma produced. An ELISpot reader can be used to identify and quantify the number of relevant spots in each well. Since it does not depend on T cell activation, staining of T cells with MHC multimers is probably the most elegant approach to detecting antigen-specific T cells. However, it requires that the precise antigenic determinant, or epitope, of the detected T cell is known before the assay is carried out. MHC multimer staining is also the most limited approach because it detects only T cells with that one exact specificity. MHC multimers consist of recombinant MHC molecules with a specific peptide bound in their binding grooves. Several of these MHC peptide complexes are joined by a backbone molecule that also carries a fluorescent tag for identification. Unlike activation assays, this method detects T cells based on the presence of specific TCRs on their surface. These TCRs specifically recognize the peptide when held in the binding groove of the recombinant MHC molecule. It is thought that this approach will identify all antigen-specific T cells specific for the MHC peptide complex, since recognition does not depend on T cell activation. For CMV, it was shown that typically around 60% or so of the T cells that can be detected by MHC multimers will produce interferon. The stimulating antigens that are used for T cell activation assays vary widely. They range from crude cellular lysates to individual, highly purified peptides. Sources of antigen include bacteria, viruses, fungi, or other antigens. The antigen format determines which T cell populations are likely to respond. Antigens that require processing by antigen-presenting cells, or APCs, are more likely to stimulate CD4 T cells because they are processed through the external antigen processing pathway, producing peptides inside phagolysosomes. These peptides are transported to the cell surface bound to class II MHC molecules, where they are recognized by class 2 MHC restricted CD4 T cells. Antigen preparations that require processing by APCs are all those that contain intact proteins or large protein fragments. For example, a lysate of CMV infected fibroblast, otherwise CMV lysate. If such antigens are used, it is important that vital APCs are present in the cell suspension to be stimulated. A fresh PBMC sample from a CMV infected individual will usually show CD4 T-cell activation when stimulated with a CMV lysate. However, this works less well with frozen PBMCs, probably because APCs have died or lose function after freezing. Antigens that do not require processing by APCs are preferable for use in T-cell activation assays because processing can be rate limiting in terms of activation and is dependent on APC viability. It is important to note that in order to fit into the binding groove, a particular peptide binding class 1 MHC might require clipping, which appears to happen outside the cells and probably by so-called clip peptidases. Please note that the only antigen format that can stimulate CD4 and CD8 cells equally, the use of peptides for T-cell stimulation has been well reported. Peptide length can vary from very short to very long. The minimum length for T-cell epitopes is thought to be in the range of 8 to 10 amino acids, matching the length of a peptide that can typically be accommodated into the peptide binding grooves of MHC molecules. We recommend peptides of 8 to 11 amino acids length for CD8 T-cell stimulation and peptides of 12 to 20 amino acids length for CD4 T-cell stimulation. There is a peculiarity about CD4 T-cell stimulating peptides. They can be slightly longer than the MHC binding groove, and the TCR may recognize residues flanking the core epitopes. It will depend on the exact epitope, which peptide length is best. Ideally, the peptide would cover the entire epitope, but at the same time, not be much longer than required for optimum recognition by the cognate TCR. With respect to class 1 presentation, if peptides are too long, they might not be able to be clipped to fit into the MHC1 binding groove. This will depend on the exact peptide and is currently not predictable. Note that peptides of the right length do not necessarily automatically bind to a given MHC molecule. This will depend on the side chains of the peptide amino acids and those of the amino acids forming the binding groove. Important positions within the binding groove are referred to as anchor residues. This slide shows you why there are differences between peptides that represent CD4 T-cell epitopes and peptides that represent CD8 T-cell epitopes. The class 1 MHC binding groove is closed at both ends, whereas the class 2 MHC binding groove is open. At each extremity, the class 1 binding groove forms pockets requiring the N-terminal and C-terminal ends of a binding peptide to fit in order for the peptide to be presented. These pockets contain important anchor residues. Class 1 MHC presented peptides are thought to be able to bulge out of the binding groove in the middle, which explains why sometimes peptides longer than 8 or 9 amino acids can be accommodated. Class 2 MHC binding peptides can, because of their processing requirements, stimulation with proteins or large protein fragments depends on an intact presentation pathway for external antigens. Antigens are processed in a phagolysosome and eventually presented on class 2 MHC molecules on the cell surface. As a result, CD4 T cells may be stimulated efficiently, but not CD8 T cells. However, proteins can be replaced by peptide pools that cover the protein amino acid sequences with sufficient overlap in order not to miss stimulating peptides along the sequence. Such peptides will be loaded onto the MHC complex externally, do not require processing, and will efficiently stimulate CD4 and CD8 T cells. JPT provides peptide pools in any requested format. However, we recommend our standard PepMix pools, which provide peptides of 15 amino acids length with 11 amino acids overlap. Occasionally, we may deviate from that pattern for peptides that cannot be efficiently generated. In our hands and those of many investigators, the 15 by 11 configuration has been extremely useful for identifying CD4 and CD8 T-cell responses, with all peptides representing a complete protein contained in one single pool. Such pools may also contain additional peptides to account for known sequence variations in the target protein. We call them ultrapools. When screening immune responses against peptides, having to make a compromise is inevitable, since not every possible peptide can be offered. Frequently, peptides are generated specifically for screening for CD4 epitopes or CD8 epitopes, in which case peptides will have different lengths. For example, 11 amino acids with 10 overlaps for CD8 T cells, and 18 amino acids length with 17 overlaps for CD4 positive T cells. If the sequence of a whole protein needs scanning, this will generate many peptides and be very costly. This is why researchers often use longer peptides using a minimum acceptable overlap. Again, this is always a compromise and will take into account a number of factors, including cost, the maximum number of peptides that can be handled, and the amount of donor material that is available. In the same way that peptides representing the same protein can be combined in peptide pools, one may also combine peptides originating from different proteins or organisms in one single pool. For example, our CEFT pool contains peptides originating from cytomegalovirus, Epstein-Barr virus, influenza virus, and Clostridium tetani. This pool is often used as a positive peptide-based control pool. A response to this pool will indicate that there is a response to one or more of the antigens represented by this pool. However, while any number of represented antigens may contribute to the response, it cannot be discriminated which, unless peptides originating from each individual antigen. Of note, once peptides are synthesized, they may be combined in one large pool. However, we also offer the possibility to keep individual aliquots in defined amounts for use in subsequent epitope identification. Strictly speaking, these methods identify T-cell stimulating peptides rather than epitopes, as epitopes include parts of the MHC molecule that peptides are presented by. Arranging peptides in smaller pools in such a way that each peptide is contained in exactly 2 pools creates a crossover matrix that allows the identification of stimulating peptides from the responses obtained within these matrix pools. We also refer to them as mapping pools. They are represented by the columns and the rows in the matrix table in the middle of the slide. The example shows a one-step identification of a CD4 T-cell stimulating peptide and its confirmation using the individual peptide for stimulation. Pools 8 and 20 gave positive responses. This identified peptide 92 as the stimulating peptide because it is the only peptide they have in common. You can also see the response with pool 21, which is a negative response. When using flow cytometry, the initial assay with the matrix pools and the confirmation with individual candidate stimulating peptides can be done in short sequence. For example, after overnight stimulation with the matrix pools, stimulating pools are identified on the 2nd day, and after a further overnight incubation, the individual stimulating peptides are identified on the 3rd day. If more than one pool across and one pool down give a positive response, there will be more than one candidate stimulating peptide, and as a result, several will have to be tested for confirmation. In our hands, PBMCs were used successfully for up to 72 hours after preparation if kept in complete media in the incubator. Using matrix pools in combination with flow cytometry may be the fastest way of identifying T-cell stimulating peptides to date. Of course, if PBMCs are live frozen, the different steps of stimulating peptide identification can be timed differently. A lack of proper external standards for T-cell assays has been limiting meaningful comparisons of results between different runs, different platforms, or different laboratories. Typically, a sample from a patient or study participant needs to be assessed. With respect to T-cell activation assays, a positive assay control will confirm that the assay has worked in principle. However, it will not tell us much about the quantitative performance of the assay in a specific run. A negative control, by contrast, will tell us if there are stimulated T cells, even in the absence of the specific stimulus. Such stimulated cells are not specific for the stimulus we use, but will occur as background. Unless we have a sample whose specific response under ideal assay conditions we know, we cannot tell if the response in the test were generally stronger or weaker than in a previous run. A sample containing T cells of a known specificity at a known frequency is required for that purpose. While this could in theory be any sample responding to a peptide, it is a great disadvantage that we cannot set the frequency of responding cells to a specific value, or create specific samples with several different values. We cannot dilute the sample within itself unless we remove T cells from the fraction we are using for dilution, but this would require complex and costly experimentation. Also, we cannot switch the reactivity of a sample and therefore would need to screen a number of blood donors in order to identify one for each specificity and frequency of antigen-specific T cells that we require. In regards to MHC multimer staining, a negative control is usually produced by using an irrelevant MHC multimer. This assesses the binding of an MHC multimer that could not bind and therefore provides a measure of unspecific binding. A positive control would be a cell suspension with a given frequency of cells to which the MHC multimer is expected to bind. The problems associated with the TERS, or TCR Engineered Reference Samples, technology provides a solution to the assay standard problem by creating individuals and bespoke external assay standards. These are cell suspensions with a defined number of T cells bearing the desired TCR. This is accomplished by transfecting TCR encoding RNA into fresh PBMCs using a quick and simple electroporation protocol. TERS kits contain linear RNAs encoding alpha and beta TCR chains for selected TCRs, for example, CMV or influenza, as well as linear RNA encoding green fluorescent protein or GFP for protocol optimization. Producing TERS with the universal T-cell assay standard TERS kit is very quick and easy. Before TERS can be produced, the electroporation-based transfection protocol should be optimized using the green fluorescent protein RNA provided in the kit. This can be done using PBMCs or other cell suspensions, but ideally the same type of cell suspension that will be used for TCR transfection afterwards. Once PBMCs have been obtained, they are split into two portions. One is used for TCR transfection, and the other is left untouched for adjusting the concentration of TCR transfected cells later on. The next step is the transfection of PBMCs with the provided TCR mRNA using the now optimized electroporation protocol. Once this step has been completed, the transfection efficiency should be determined using MHC multimer staining and flow cytometry. It is typically around 60 to 80%, which means that 8 out of 10 CD8 T cells bear the transfected T-cell receptor. Now it is time to adjust the desired frequency of TCR positive T cells by diluting the transfected PBMCs with untouched PBMCs. You may want to have 0.1%, 1%, or any other frequency of your transfected cells in your TERS. You may also want to have two preparations, for example, a high and a low control. It is wise to confirm the TCR-bearing cells are present at the desired frequency in the final cell suspension using MHC multimer staining and flow cytometry. Finally, TERS are frozen down in suitable aliquots for later use. The kit contains a detailed. Once you have a TERS with your preferred specificity, you may use it to monitor the performance of your assay from one run to the next. You may use it to calibrate your assay or simply explore its detection threshold. You can produce enough TERS samples to permit running the same controls every week for years. You can also use TERS to compare the performance of the assay between different people in the same or in different labs or under different conditions. When you run out, you produce more with a fresh PBMC sample, and you can adjust the frequencies again to what you require. The benefits of TERS are obvious. TERS provides reliable external assay standards for an antigen-specific T-cell assay. TERS achieves a new level of quality control. TERS enables studies to run across platforms and laboratories. For additional information on this and other innovative peptide solutions from JPT, please visit our website at www.jpt.com, or to reach one of our experts, you may write us worldwide directly at info@jpt.com.